strand. And in the endoplasmic reticulum, the membranous sacs are connected to one another. And that's what distinguishes the endoplasmic reticulum from the Golgi apparatus. Both of them are made up of these flattened membranous sacs that are called cisternae. But in endoplasmic reticulum, the cisternae are interconnected, while in the Golgi apparatus, to one another. So they are simply stacked close together but not connected. Now the endoplasmic reticulum includes a rough and a smooth variant, and so we describe the rough endoplasmic reticulum and we describe the smooth endoplasmic reticulum. The rough endoplasmic reticulum is made rough by the presence of ribosome docking proteins. Ribosome docking proteins are anchoring sites for ribosomes, and we say that ribosomes are a molecular protein printer. Right? I'll say that over and over and over. Yes? Does anyone have a whiteboard marker? Nuclear envelope. And that suggests to you that it has a critical role in protein synthesis. The smooth endoplasmic reticulum is further away from the nucleus and is involved in lipid metabolism. We're going to talk about that a bit. So you'll be taught about the process of protein synthesis in physiology. The instruction arrives from wherever, right? From an intracellular source or an extracellular source. The instruction arrives to increase protein demand. These notes are available on the mega folder, so you don't have to take pictures. Begins when a request for gene transcription arrives in the nucleus. And the request simply says, activate this section of the genome so that the proteins it codes for are produced. Right? And what happens is the DNA is uncoiled and an enzyme reads the sequence of nucleotides. As it reads the sequence, it generates a complementary mRNA sequence. And so it's essentially making a copy. Right? When the copy is made, the copy is released into the nucleoplasm, right, into the fluid of the nucleus, and it's tagged for transport outside the nucleus. And so it floats out of the nucleus, passes through a nucleoporine and enters the cytoplasm. When it enters the cytoplasm, that strand of information is attached to a ribosome. And the ribosome starts to read the sequence. Now you remember that every 3 nucleotides, every triplet in the nucleotide chain codes for an amino acid. Yeah, you remember that? Great. So the ribosome reads a triplet, and meanwhile, within the cytoplasm, you have what are called tRNAs. And tRNAs have a complementary triplet for each amino acid. So a tRNA attaches to an amino acid and it has the complementary triplet. So that if this is GCT, this is an mRNA, so you don't have C. You don't have T, you have U. So if this is a GCU, you have a tRNA for A, U, C, right? And this codes for the same amino acid as that tRNA. So the ribosome is reading the mRNA sequence, and as it reads the mRNA sequence, it brings the mRNA and complementary tRNA together, and it holds them in place. The tRNA is holding the amino acid, right? And then it moves on to the next triplet, and it reads the amino acid code, and it brings the amino acid next to the previous one. And these 2 amino acids are joined to one another. And the ribosome travels down the mRNA strand doing that. bringing in the tRNA so that the amino acid chain is created, right? And then, once you have a long enough chain, the chain is fed into the rough endoplasmic reticulum. So typically what happens is the ribosome attaches to the rough endoplasmic reticulum and the chain is fed into the cavity of the rough endoplasmic reticulum. And within the cavity of the rough endoplasmic reticulum, the chain undergoes, first it completes synthesis of the polypeptide chain, and then it undergoes post-translational modification. Now, post-translational modification can involve many things. So you can cut out unwanted portions. You can fold the amino acid chain so that you bring amino acids together, right? And so if you want to form a sulfide bond inside a protein, you bring two amino acids with sulfide bonds together with sulfur molecules so that you make a sulfide bond. And so those are processes of post-translational modification. You can add molecules to this newly made protein. So do you want to add a sugar, right, or a carbohydrate molecule to it? Do you want to add a carboxyl group? Do you want to hydroxylate this protein? And so all those things happen inside the Golgi apparatus. And then once that's, inside the rough endoplasmic reticulum, once that is completed, the protein is passed on to the Golgi apparatus. And the Golgi apparatus performs some post-translational modification, but mostly it packages the protein, it addresses it to its destination, so whether this protein is destined for the plasma membrane, whether it's destined for the cytoplasm, or it's destined for another organelle. So for example, lysosomes contain enzymes, and those enzymes are made by ribosomes and inside the rough endoplasmic reticulum. And when they finish packaging inside the Golgi apparatus, they are addressed to be sent to the lysosome, right? And so you address it, and then the Golgi apparatus finally dispatches the package. And so it takes the package and hands it over to one of those proteins we talked about in the morning. We call them molecular motors. So it hands over the package to a molecular motor, and the molecular motor carries the package as it travels along a cytoplasmic filament to the destination. And so that's generally the process of protein synthesis. You'll repeat that in physiology, and you'll repeat it in biochemistry, and they'll emphasize on the relevant bits. I'm an anatomist, so I don't know protein synthesis, yeah? Now, the smooth endoplasmic reticulum is involved in lipid metabolism. So most lipids that you take into the cell will be processed in the smooth endoplasmic reticulum. But it's also involved in detoxification. And so when you take, there are molecules that we take into our bodies that are physiologically active. Those molecules are called xenobiotics. So a physiologically active molecule that came in from outside the body is known as a xenobiotic. And the smooth endoplasmic reticulum contains the enzymes that break down xenobiotics, right? Xenobiotics can be anything from medicines to plant compounds like caffeine, right? To alcohol. All those will be broken down inside smooth endoplasmic reticulum. And so tissues that perform a lot of detoxification will have a lot of smooth endoplasmic reticulum. All right, and then the smooth endoplasmic reticulum is the site of glycogen metabolism. A really important function that we don't talk about enough is membrane formation. And so plasma membrane must be synthesized somewhere. It doesn't just materialize out of the blue, it must be synthesized somewhere, right? When a cell cleaves, it takes half of the plasma membrane of the parent cell, right? So that's what happens when cells separate. They take plasma membrane from the parent cell. But exocytosis and endocytosis are constantly depleting plasma membrane. And so you need to keep regenerating plasma membrane and recycling what has been taken into the cell during endocytosis. And the smooth endoplasmic reticulum is responsible for that process. And then finally, the smooth endoplasmic reticulum is a site of calcium storage. And it becomes very important in tissues that store large amounts of calcium like muscle. And so smooth endoplasmic reticulum in muscle is specialized for the storage of large quantities of calcium and is called sarcoplasmic reticulum. But we'll talk about that when we talk about muscle tissues. Alright, the Golgi apparatus is involved in post-translational modification of proteins, packaging and dispatch of cellular proteins. And you can see that the Golgi apparatus is just external to, you can understand such terms, right? External, internal, Mnarada, so I can use them freely. So you can see that the Golgi apparatus is just external to the rough endoplasmic reticulum, and the reason is because it's receiving that incompletely modified protein so that it completes the process of modification and then it dispatches it, right? It packages it in a vesicle, it addresses the vesicle for the destination and it sends it out. And the proteins are packaged for dispatch to the apical membrane, the basolateral membrane, endosomes and lysosomes. So you're sending them to various destinations. And the idea of addressing those proteins is critical because apical proteins are different from basolateral proteins. And so you don't want the wrong proteins going to the wrong site. You'll have a defective cell. Lysosomes contain various enzymes involved in the degradation of biomolecules, pathogens, and worn out cell organelles. So this is an illustration of a lysosome. A lysosome is one of the only cell organelles after the nucleus that has a bilaminar membrane. Right? And so the membrane of a lysosome actually has two lipid layers. And the lysosomal membrane is impermeable to lysosomal contents. So the things inside the lysosome never leak outside the lysosome. For anything to enter or leave the lysosome, it must pass through transporter proteins. Right? And the reason for that is because the lysosome is digestive. And if the lysosome were to empty its contents into the cell, its contents would digest the whole cell because the cell is lipid, protein, carbohydrate, and nucleotide, right? And the lysosome contains proteases, lipases, carbohydrases, and nucleotidases of various types. So lysosomes are formed by insertion of lysosomal proteins into endosomes. So what happens is the proteins are formed in the rough endoplasmic reticulum sent to the Golgi apparatus. And in the Golgi apparatus, they have to be packaged in an impermeable way. And then that packet fuses with a membrane vesicle called an endosome. When they fuse, you form a lysosome, right? Yeah, that's about it. And lysosomes are also involved in autolysis of cells during programmed cell death. So a severely injured cell undergoes rupture of its lysosomes, and the lysosomes auto-digest the cell. But typically, we have more efficient, less destructive mechanisms of cell death. One of them is called apoptosis. And during the process of apoptosis, you generate an enzyme that simply chops up the DNA. When you chop up the DNA of a cell, that cell might as well, it is dead, right? So you chop up the DNA into these nonsense sequences, and the cell simply disintegrates into tiny, tiny packets that can be mortar, right? Okay. Mitochondria are familiar to all of you, yes? Yeah, fantastic. They have this outer membrane, right? They are sausage shaped, sometimes they are spiral shaped, sometimes they are round, sometimes they are filamentous. Mitochondria occupy various shapes, right? Based on the cell that they are inside of. But the thing that distinguishes them, we can tell ourselves a story. Yeah? So, a long time ago, when the first eukaryotic cells were forming, so the earliest cells were photosynthetic cells, right? They relied on the sun, and actually, like at the earliest points in time, you don't have a cell. You have different membrane-bound units that sustain themselves by doing different things, right? And what happens over time is they start to form symbiotic relationships. Yeah, and so the earliest algal cells, the earliest algae cells, were really inefficient at photosynthesis until they met a highly adapted organism called a chloroplast. And they entered a symbiotic relationship with the chloroplast, and over time the chloroplast became part of the plant cell. So the same thing happened to eukaryotic cells. The earliest ones were really inefficient at energy production until they encountered this independent unit floating around in that early primordial soup. And they entered a symbiotic relationship with that organism. And eventually that organism became a part of the cell. But mitochondria have always, mitochondria do their own business. Yeah? For the most part, they're independent of the cell. And so mitochondria increase and decrease in number based on what they want. Yeah? They replicate on their own terms. In fact, and this one is interesting, all mitochondria in your body came from the ovum that was fertilized by the sperm, right? And so the ovum, on top of providing a cytoplasm that facilitates development of the zygote, actually contributes the mitochondria that will be found in the cells of the organism. And so your mitochondria are like your mom's mitochondria, which are like your grandmother's mitochondria, which are like her mother's and her grandmother's, and on and on and on. And because almost all mitochondria, because mitochondria are highly conserved, right? Then you can actually group human beings based on mitochondrial lineage. And so we all belong to a small number of mitochondrial families, right? And if you walk far back enough into the archaeological record, you can get to the progenitor who gave rise to all the mitochondria inside the cells of man. Yeah? And so the oldest ancestor of human beings is actually that person, right? Who all mitochondrial families are linked to, and we call her mitochondrial Eve. Yeah? So that's the oldest verifiable ancestor of man. All others are stories. Anyway, back to this cell. So it has this outer membrane, and the outer membrane is freely permeable to substances. So that the intermembral space, the space between the outer and the inner membrane, that intermembrane space, is similar in constitution to the cytoplasm. So it has the same solute concentration, it has the same nutrient concentration as the cytoplasm. The inner membrane, so the other thing that's found inside that intermembrane space is the earliest enzymes of the mitochondrial chain. The inner membrane is impermeable, or at least it's selectively permeable. It allows the nutrients that we break down to go in, but it allows hydrogen ions to flow only one way. Right? So the nutrients go in, but the hydrogen ions can only flow along a concentration gradient out of that inner space, which is called the matrix. And so the purpose of the enzymes inside the innermost space is to generate hydrogen ions. Yeah, that's like the big deal. A mitochondrion generates hydrogen ions and accumulates them inside the matrix. Once you have a high enough concentration, you create channels for those hydrogen ions. And as the hydrogen ions flow through it, that protein behaves like a turbine. So you know how you build a dam, you accumulate a large amount of water, and then at the bottom of the dam you stick a turbine, right? And as that tall column of water pushes the water through the turbine, you rotate the blades of the turbine, and that rotary energy is what we harness to create electricity, right? And so the same thing happens in mitochondria. You accumulate hydrogen ions, and then you let them out through these turbine-like proteins. And as they flow out, they give you the energy that you use to take adenosine diphosphate and attach it to a phosphate molecule. And that last phosphate to phosphate bond is what we break everywhere else to generate energy. And so the mitochondrion generates the ATP, and the ATP diffuses into the cell. And then every time you need energy, everywhere else in the cell, you simply break that phosphate bond and it releases a small packet of energy that you can use to perform activity. So again, biochemistry and physiology will explain that stuff to you. So, alright, we are done. So, mitochondria are also involved in apoptosis. So, you remember I told you that for apoptosis, the body relies on an enzyme that chops up DNA, right? So, that enzyme is actually contained inside the mitochondria. And so, cellular injury triggering apoptosis must damage mitochondria. And once mitochondria perceive a critical level of cellular stress, they release that enzyme into the cytoplasm. That enzyme is called caspase. And caspase floats through the cytoplasm into the nucleus and it chops up the DNA and turns it into nonsense. And then the cell is dead. Peroxisomes are a type of organelle involved in the elimination of reactive oxygen species and fatty acid oxidation. And so they perform detoxification and they also perform lipid metabolism. They break down lipids to generate energy. So we talked about reactive oxygen species in our last classes. Did we? Do you remember? Oh yeah, and I tried to ask you to remember what we talked about last week and I couldn't. And that's okay. That's alright. That's all right. So they're involved in the elimination of reactive oxygen species, what we call free radicals, right? And we talked about the dangers of free radicals. Does anyone recall? Yeah. I had a yes. Oh yes. Baba Jimmy alikuwa anasema hivyo. Unajua Baba Jimmy? Oh yes. Oh yes. Yes, I had a yes. On the cells, how? I know you remember. Don't be scared of these guys. Okay, do you remember? Okay. So free radicals. So in this case, specifically free radicals of oxygen, right? They're what we call reactive oxygen species. An example is an O2 molecule with an additional electron. Yeah, so it will be O2 negative. Yeah, or an oxygen that is not bound to another oxygen, in which case it will be an O2 negative, right? And so it's walking around with this electron that is unbound, and it is looking for any other molecule to bind to that electron. And most of the molecules it will encounter will be plasma membrane molecules, right? And when it alters the composition of the plasma membrane molecule, it affects plasma membrane integrity, right? When they are inside the cytoplasm, they are affecting the integrity of all the organelles. And so it's important to get rid of them as quickly as possible because they are going to disintegrate your cell, right? It's what, have any of you ever gone to hospital and a wound that you had was cleaned with hydrogen peroxide? Yeah? Now, hydrogen peroxide very quickly breaks down in the presence of air into water and an oxygen radical. And that oxygen radical saponifies your tissues. It turns them into soap. And so there's a... Yeah? It sort of cooks your tissues in real time. Ukiona tu unaiva. And so that's the sort of stuff you're dealing with inside the cell. And you need to get rid of it. Otherwise, inaivisha cell zako. And so that's what a peroxisome does. Yeah, and it does that through the activity of two enzymes. Yeah, so at least these are two notable enzymes, an enzyme called catalase and an enzyme called peroxidase. And both of them break down hydrogen peroxide and similar molecules. Yeah, and so because some cellular processes actually generate peroxide as a byproduct. And then finally, the cytoskeleton is involved in cellular support, cellular transport, and cellular motility. And the cytoskeleton is made up of 3 types of filaments. So you can go check out that table later. If it doesn't make sense, leave it. Kikata washana nae. But we have 3 types of cytoskeleton filaments. You have microfilaments, the smallest type of filament. You have intermediate filaments and microtubules. Microtubules have the largest diameter, microfilaments have the smallest diameter. And then intermediate filaments lie somewhere between microtubules and microfilaments in diameter. The microfilament is an actin polymer. And so in the body you have, the molecule is called actin. Yeah? So an individual actin molecule polymerizes with others to create a filament of actin. And that actin filament requires energy for its synthesis and its reconstruction, and it's in a state of dynamic instability. So as it grows, it degrades on the opposite end, right? And one of the implications of that is you can increase its length and you can decrease its length. Yeah? Now, in the body, because it's always in a state of dynamic instability, then one of the things the body does is it stabilizes actin filaments using proteins. And actin filaments are typically found just underneath the plasma membrane. So just underneath the plasma membrane, in most cells, you have a shell of actin filaments tied together in a web-like structure. Mshaiona zile lampshades zimetengenezwa na uzi. Yeah, you've seen them. So someone soaked the uzi in starch, and then they got a round balloon, and they wrapped it with that dyed uzi. Alafu ikikauka, you just pop the balloon. And what you're left with is a mesh of thread in a spherical shape, right? And so actin is sort of like that, right under the cell membrane. It's called the cell cortex. Now that allows actin to form an anchoring site for the cell with other tissues, right? And so you have proteins outside the cell, structural proteins outside the cell, and you have actin inside the cell. And if you link actin to those structural proteins inside the cell, you've created a pathway for force to be transmitted into the cell. So that if you pull from the outside, the actin deforms. And that allows your cell to change shape. Right? Make some sense? Kinda, maybe not. Okay. Should I try again ama nisonge tu? Okay, let's try again. Najasi tumebeba maka, so sijui tutasaidiana aje. Kuna msee akona maka? I've asked this before. So, to imagine, you guys know how to work with balls. So you know that the ball has the outer cover, the lever, right? And just underneath the lever is the tube. And so you have a layer of actin mesh. So those filaments woven together just under the cell membrane. So if the membrane is that outer cover, then the cell cortex is that tube right underneath the cell membrane. And because the cortex is anchored to proteins outside the cell, and it is also anchored to receptor proteins that attach to structural proteins outside the cell, then the cell cortex can respond to tension coming from outside the cell. Does that make sense? Yes. Yeah, so if it's anchored to the membrane and something in the membrane is anchored to something outside the cell, if you pull on the thing outside the cell cortex responds. And so the cell cortex allows cells to change shape. And so for example, when a white blood cell wants to extend pseudopodia, you have a receptor that anchors to a structural extracellular protein. And then the receptor on the inner side of the membrane is anchored to the cell cortex, and it changes the shape of the cell cortex, right? But then the cell cortex in some cells, so in cells that have microvilli, the cell cortex extends filaments of actin into the microvilli to help the microvilli maintain their shape. And that's a lot of what actin filaments do. The other place we find actin filaments is inside skeletal muscle. So in skeletal muscle, actin and myosin interact to shorten skeletal muscle cells and cause contraction. The other filament type is intermediate filaments. Unlike actin filaments and microtubules, intermediate filaments do not exhibit dynamic instability. So once an intermediate filament network is created, that network tends to be stable. And so inside the nucleus, for example, the filament that helps maintain nuclear support and shape is an intermediate filament. Inside a cell like the neuron, and so neurons are really long, right? And so you can have a neuron extending from your lower back all the way to the tip of your big toe. So in such a cell, that long cytoplasmic process contains inside it intermediate filaments so that it does not collapse or kink or fold or knot on itself. In places like the skin, actually most epithelia, where you need some resistance to friction, then you have keratin, which is an intermediate filament, right? And it's deposited inside those cells and it accumulates there. And because it doesn't break down easily, it can last a long time, right? And so keratin inside a skin cell will last for the lifetime of the cell. That's like maybe 70 days. Okay, actually it's maybe 7 weeks, around 49 days. Until the cell is shed and another one takes its place that's also full of keratin. And so intermediate filaments are the most stable filaments inside the cell, and they have a really important structural role. And then finally you have microtubules. Microtubules grow and regress, right? They also exhibit dynamic instability, and so we rely on proteins to help them retain their shape. Microtubules in the cell are important because there are other proteins that can travel on microtubules. And so you remember I told you about these molecular motors that can be loaded with cargo at the Golgi apparatus, right? Now those molecular motors have arms that hold cargo, and they have legs. And the purpose of the legs is to walk along the microtubule. And so the microtubule is built from its origin towards its destination, and the molecular motor moves along that microtubule. When a cell is undergoing mitosis, the duplicated chromosome, right, occupies what is called the cell equator. And attached to the centrosome of the duplicated chromosome is a molecular motor called a kinetochore. And that molecular motor attaches to a microtubule. And once it does that, the reason the chromosomes separate is because that molecular motor starts to migrate along the microtubule towards its origin. And so you end up with the halves of the chromosomes moving towards opposite ends of the cell, right? And so microtubules are important for transport, and they're also important during the process of cell replication. And if you fail to form microtubules during cell replication, the cell dies. It undergoes what is called a mitotic catastrophe. And over time, what we have learned is ways to impair microtubule assembly. And so some of the medications that we use in cancer to treat cancers actually impair microtubule assembly, and they cause mitotic catastrophe inside cancer cells. And so those cancer cells no longer replicate, and the tumor mass shrinks. Yeah? All right. So that's sort of it about the cytoskeleton. You can actually go check out the table. Ile utaelewa tu, ile itapita uachane nayo. I think the location in the cell might actually be one of the most important bits. Now, you have microtubules inside structures called cilia. And cilia are extensions of the apical cytoplasm. They are finger-like projections, just like microvilli. But the difference between cilia and microvilli is that cilia are freely motile. And so cilia actually move. They use energy to move. And as they move, they can sweep material on the surface in a particular direction. So for example, in the respiratory system, all cilia beat towards the pharynx. The cilia inside the nose beat posteriorly towards the pharynx. The cilia in the lungs beat upward towards the pharynx. And this is why when you don't notice, for the most part, but you're always producing mucus in your respiratory system, and the mucus is being swept into the pharynx and you swallow it. You only notice when the rate of mucus production is so high that first, it can't all be swept up, and second, it's a lot, and so you feel yourself swallowing. And so when you have an infection, for example, in your airways, you cough because the mucus has exceeded the ability of cilia to carry it towards the pharynx. And so you're coughing to help remove that mucus. Yeah? Anyway, so that was just about cilia. A cilium has the same structure as the flagellum of a sperm. And so the core of a sperm flagellum is made of microtubules. Cool. Yeah, so actually, if you just look at this last table, it might be the most informative one. Oh, we're not done. Oh, guys, you thought we were done? Oh, wow. Kwanza sasa hivi ile kumenyesha